Great, thanks, Mike, and thanks, Dan, for the kind of that opening talk and really talk about how the interface works. Brent and I are going to talk a little bit about how the model was developed, and then also some of our ideas for improvements that we're looking to do in the future. The project has really been going on for quite a long time, and I want to give a shout out to my Virginia Tech colleagues, particularly Keith Yoder and Leon Combs, who really did the lion's share of the original work on the model. Both have now retired, and lots of other people, you can read the names there, who have been involved in this project. There's others who are also testing and trying to validate this in different parts of the country. We've gotten a lot of funding from Washington State for this project, especially in the early years. They do a lot of bloom thinning out there, and it was really important to them. And that also will inform you about the varieties that we have in the model right now, which were the ones that were important to them at the time. But also funding from the Virginia apple growers, the New York apple growers, and a fairly new, I guess it's been out now for over a year, so it's not so new, but USDA Specialty Crop Research Initiative project that's headed by Terrence, called Pac-Man Precision Apple Crop Management. So clever name on that one. Okay, so my appointment is teaching and research. I know I do a little bit of extension. So as a teacher, I always like to ask pop quiz questions. So I am going to ask you guys to answer this in the chat while I go through the talk, and then we'll revisit this question later. So how many fertilization events are needed to set a full crop in a high density apple orchard? Okay, so you can throw out some guesses or figure out what that might be, and then I'll come back to it and give you my assumptions on that. So we have not a lot of history with bloom thinning in New York State. It's kind of something that more and more growers are looking to do, but historically has not been a common practice for crop load management. But there's a lot of pros, right? You get larger fruit because you're thinning earlier, and you have less competition during that cell division phase, right? When fruit size really can be increased by having more cells. A lot of our research has shown that you get greater return bloom when you do bloom thinning versus thinning later. And that's again because we are preventing seeds from forming, and those seeds produce chemicals that inhibit flower buds. So we can help to break some of the biennial bearing cycle by early thinning. Possible reduction in fungicide applications. We did some work with lime sulfur and regalia for that. Or some of these materials are available for organic growers, where the materials available at the petal fall 10 millimeter up to 25 millimeter are generally synthetically derived and not available for organic. And now we have this model, this what we call the pollen tube growth model, which allows for more precise applications. In the Eastern US, though, I think some of the reasons why it has not become a widely used practice is because of frost primarily. And so that's been a really big issue for a lot of growers, and still is. Every year we think about that and how that might affect bloom thinning. We haven't had a lot of experiences with it, and a lot of the timing was fairly subjective based on the percent of full bloom open, 20%, and then a second application at 80% was the old rule of thumb with this. It worked sometimes, it didn't work other times, and I think it was because it wasn't based necessarily on the biology of the plant. It was based on just a visual, how many petals are you seeing in an orchard. And the materials that we have for bloom thinning tend to be caustic and can cause things like russeting. So just a little reminder on apple flower anatomy. Here's our apple flower here on the far left. Here's kind of a cross section through. So what we're interested in for the pollen tube model is the green section here, right? This is the female part all the way down through the ovule here. And this is where our pollen comes from. These are the anthers and the filaments. These are the pollen source, that's the male. What we're interested in is when the pollen lands on the surface, and we'll look at it on the far right here, of the anther, right? So pollen is dry, it has to hydrate, there's a lot of protein and some molecular reactions, and then it germinates and you get this pollen tube growing down through the flower. Here's the base of the style. here's our ovary and our ovules, and inside the carpel, and the pollen tube needs to grow in there. And then there's a double fertilization practice or event that happens, and then you have fertilization. Okay, so the pollen tube growth model is measuring the time it takes from pollen to start growing on the top of the anther all the way down to the, the top of the stigma, all the way down to fertilization. Okay, and we figured this out through a lot of empirical work. We had these dwarf apple trees, kind of bonsai apple trees. They were in the field like this over here in the top right. And we can pull them out of the ground because they're in root bags, and then force them in a greenhouse to flower, emasculate them, do controlled pollination, stick them into a growth chamber that we're seeing here on the bottom left picture at a set temperature. And then after a certain amount of time, pull them out, harvest the flowers, and then we can visualize the flowers using fluorescent microscopy. So this is, again, some of the pictures on top. This is very similar to what a breeder would do, right? They're taking pollen and they're just putting it right on top of the stigma. And then this is some of the visualization here. So these are the pollen tubes on the top here. You can see them fluoresced in blue. And then this is just pubescence down here. And then these darker blue lines are measuring. This one is 8.3 millimeters. Okay, so by doing these at different temperatures in the growth chamber, we can measure the growth rate, right? If you know the length by time, we can figure out rate, and then we do this at various temperatures, and so then we can generate these curves here. And then if you then take this out into how does this apply to what you're doing out in the field, we have all these weather stations with NUA now, so they're recording hourly temperatures. So the data gets sent to NUA into their servers. They use the equations that we built through this model, and then they can generate the amount of growth of the pollen tube each hour, and then add them up so you get this cumulative growth curve over time. Okay, up to 100%, which is the style length. Okay, so the style length is something that you have to measure in the field, and so we're measuring from the top of the stigma to kind of through the middle of the carpel there, almost down to the calyxes here. So not just the styles themselves, but down to the bottom there. And we figure that fertilization is probably happening somewhere in there where my pointer is. So we are measuring the starting point to the finish line, and how long does that take to happen? And that's the pollen tube growth model. Okay, and so this is just an example here of measuring. It can be measured with calipers. It can be measured with a little ruler. We generally go out and harvest a whole bunch of flowers, like 50, and measure them, and then input an average of 50 styles. Now, the other big thing is to know when to start the clock. And so that's when the last flower that you want to be your crop for that year has opened, last king bloom flower, right? The last one that is open. So that can be based on a number of metrics. Now growers are getting more used to counting these things through branch cross-sectional area like you do for fruit counting. And so if you knew that you wanted 6 apples per trunk cross-sectional area, then that could be your target. You have 6 flowers per trunk or branch cross-sectional open. Based on experience, a lot of growers are now kind of eyeballing it. I think we need to get to better systems for that, and we have some work along those lines. And then the first thinning spray happens when the pollen tubes have been modeled to grow beyond the longest style, or the average style is what you input. Okay, in other words, the flower at that point has been fertilized. And then additional thinning sprays are used to prevent fertilization. Okay, so that's the key. So they have 2 different functions of those, of measuring the pollen tube growth. The first is to allow a certain percentage of the flowers to get fertilized. And then the additional ones are to prevent any more fertilization to have happen. So this is just kind of an example. Dan showed you one. I think Michael probably showed you some. So this is cumulative growth rate of the pollen tubes up to 111% of the style length. And then we put on a caustic material and then we started the clock again, and then at 70%, we put a second spray on, and that would have prevented any more pollen tubes from reaching the base of the style and fertilizing the eggs. So we have models now for 7 cultivars. We're looking to add more. Here's our list here, and you can see these are really Washington State. We know we have important for Washington State. We know we have a lot more diversity here in the East. So we're working on that, and that's some of the work that Brent is working on. But we have been using the model for years now in Washington, Virginia, Pennsylvania, North Carolina, and of course, New York. This is an example of Washington's there. You know, there are some model limitations. It assumes that we have optimal bee activity. It's on the other slide as well, about 50 degrees Fahrenheit for good bee activity for the European honeybee. We don't have these models for all the different cultivars that we grow, or varieties that we grow in the Eastern US. We're still figuring out what chemistries to use, and I think that's you know, a big open question. I think Mike's going to show some work on some trials that he did in the Champlain Valley. And then we've done some work looking at the paternal effects of pollen sources. So the different, the model was using snowdrift, which is a common pollinizer in Washington State. We have a lot more diversity of pollen sources here in the East, and there's some questions around that. There's actually some cool research showing that when you have more than one genotype on that land on the stigmatic surface here, that they actually grow faster. They start competing with each other than if you just had one genotype. So there's some factors that we just haven't really figured out how to model yet. Okay, oops. So let's go back to the question here. And I only see a few responses in the chat of how many fertilizations we can have. I don't know, Mike, are there any there that I'm missing?
Nope, I see just those couple there. So
okay.
If you want to get your guess in, now's the time.
This is not a very interactive group. You guys are not going to get a high score in your quiz, but here's my math on it. And I just kind of scratched this out. I don't know if this is exact, but just kind of a fun thing to do. I assume that there were 1,000 trees per acre in a spindle type planting. You need about 100 apples per tree to get your 1,000 bushels per acre, you know, you go into all the different sizes, but that's a good round number to use. We figure there's 5 seeds per apple. There can be up to 10, right? So, and then we have the double fertilization event for angiosperms, okay? So if we do the first 3 bullets here, we get to 500,000 pollen tubes, and then you have 2 fertilization events for angiosperms. So then we get up to 1 million, right? That's just to get to the desired crop load, not including all the extra fruit that got fertilized. And then that's fertilization, not pollination. I don't know how many pollen grains would land on a stigmatic surface in an apple orchard. Might be 10, might be 100 times more. But I think the point is, when we're talking about modeling, you know, we're trying to model what's happening a million, several million times in an orchard, and trying to come up with a model that works for that. So, you know, some of the difficulties and challenges and the fun part of it. Okay, I'm going to pass it off to Brent now. And Brent, you'll just have to tell me to advance when you're ready.
Yes, sounds good. All right, so I just wanted to jump in here and kind of talk about the work that I'm doing as part of my postdoc project, which sort of builds off of the limitations of the current model that Greg mentioned in his talk. So this work is part of a national SCRI project that Greg mentioned on precision crop management in apples. And sort of the goal of what I'm working on is how can we use the most recent techniques in modeling, artificial intelligence, and machine learning to have a more powerful pollen tube growth model. And by that, I mean, how can we have more accuracy, more adaptability to the model, a model that is easily expanded, but most importantly, we're looking for something that's quote unquote universal. So I could go to the next slide, Greg. So initially, what I'm doing right now is taking the original data that Greg mentioned that's been collected from essentially 2005 to about 2017, and using some different techniques to do some different things with the models. So the first thing I'm looking at is different model techniques. So things like making a thermal time model, which is like a growing degree hours model, which assumes that pollen tube growth can't happen below a certain temperature. And also looking at something called the percent styler penetration. So this is, instead of looking at what the growth rate would be at given temperatures, it's basically how much the stigma is being penetrated by the pollen tubes. Next, I'm looking at some classification models. So these are models that separate or rank different factors, and this is allowing us to find which factors are going to be most important on growth rates. Is it temperature? Is it paternal pollen, or is it paternal cultivar? Is it location? These types of things. And it could also allow us to group different cultivars into ranges like slow, mid, and fast. And this might make it easier for us to add new cultivars in the model. So if we have a cultivar that we can classify as a slow, we can use the slow models as sort of a proxy. And then lastly, we're looking at forecasting machine learning models. So these are models that look at many different factors to predict a Y value. And so this would be a model that we're currently trying to develop using many different data sources to sort of train the model, so to speak. All right, next slide, Greg. So here's a look at a couple, a comparison of sort of the current model versus some of the models that I'm working on. And so on the left is essentially what we have that we're working with in NUA. So on the y-axis, you have the pollen growth rates, and then on the x-axis, you have the temperatures. However, then on the right, what we're looking at is a model that is set at a growing degree hours with base temperature of 4.5 Celsius. And on the y-axis, we're looking at the percent styler penetration. So this is just a little bit more of a transferable way for us to look at the data. So we can take data from many different experiments and create a model that has many different data points to look at many different factors and have ostensibly a more accurate model. All right, next slide, Greg. And so just a little overview of what we're doing now with the field trials to expand and validate the model. Currently, we're developing a sampling protocol that we can use in regional testing that's going to be done this spring in New York, Virginia, North Carolina, and Washington. And then we're going to do some field trials that are going to be conducted regionally to validate the model as the old models and the new model. And then we're going to take all that data, feed it into the model to expand the model to look at different factors and add more data points to make the model more accurate. So then one more click, Greg. And so hopefully we're going to have a beta testing of the new model tentatively sometime in maybe spring of 2024.
Great. I think we are at the end of our slide deck.